Hello. Am I audible? Good evening to the students who have joined. Can you please let me know if my voice is audible to you? And also let me know if you can see the presentation, the PPT clearly. Please let me know if I am audible and also let me know if you can see the presentation. Okay, thanks for confirming. Kindly wait. We will start at 6 p.m. Let more students join. If you have any doubt or any query from our previous class, which was our week 7 live session, you can let me know. Or any other topic regarding whatever we have completed till now. If you have any query, please let me know now, okay? We will start at 6 p.m. Till then, kindly wait. Okay, so it's almost the time. So I think I will be starting with the introduction part. Just let me wait for a couple of seconds. Okay, so it's 6 p.m. and let me start today's session. Wait. I hope my voice is audible to you and the screen is clearly visible. If not, please let me know. So, very good evening to the students. I'm Mousumi Deb from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I'm your TA for this NPTEL SWAM course of Medicinal Chemistry, and today it is our week 8 live session. So, before starting the session, like always, I would like to clarify a few things that this kind of problem solving session will be every week. Since it is a 12-week course, so we will be having 12 problem solving session each week. Okay, and each problem solving session will be of 2 hours, and the sessions are on Monday from 6 to 8 p.m. Okay, after completing the 12 classes, we will have one more summary session regarding the date and time that will be communicated to you after the ending of the this regular classes. Okay, all the sessions are live on Google Meet platform. This is the link for the class, and you can use the same link for joining the every Monday class, which is from 6 p.m. If any network disruption happen from my side, do not leave the session immediately. I will try to rejoin as soon as possible. I am recording the session, that is why I have muted your microphones and cameras, but the chat box is always open. You can post your doubts, your query, everything through the chat box. Okay, and if the doubt persists after class, you can drop an email to me or write your doubt in the discussion forum of the SWAM portal. I would I would happily answer your queries. Okay, and this presentation and class recording will be shared to you after the class. Since I'm recording the session, I have muted our microphones. So the class is always open. If you have any problem, network issue, you can rejoin at any time. Okay. Otherwise, this presentation and class recordings will be uploaded to the SWAM portal. So where to find these things? I have already shown it a couple of times, but let me show it one more time. So to access the class material, the recording and also the PPT, you have to go to the SWAM portal. Okay. The announcement page, I think it is visible. So on the medicinal chemistry, this announcement page, you will find there is a, on the left panel at the end, you will find there is a problem solving session 2026, right? If you click on that and if you click the problem solving session recording, you will be redirected to a page which looks like this. If you click here, you will be taken to a page which looks like this. Here you have all the date wise all the YouTube link for the class recording, and also there is a presentation folder which is a Google Drive link, and that Google Drive contain all the PPTs whatever I have we have done in our classes till now, all are in this particular Google Drive. Okay, after each and every class, generally I try to upload that material that night itself, and sometime due to some issue, if it is delayed, it will be given to you on the next day. Okay, so you can just check it tonight or tomorrow, you will find the materials. Okay, so I think the things are clear about the class and session and everything. Okay, so today it is our week 8 live session, right? So in our previous class, in our week 7 live session, we have discussed about the drug administration, the methods of drug administrations, the bioavailability, how to calculate the bioavailability, and the similar parameters regarding the pharmacokinetics and dynamics. Okay, so today let us first see what are the topics of the week 8 live session. So the week 8, we are going to majorly discuss about optimizing the drug target interactions. Okay, so whatever we have learned till now, that regarding the drug designing and so on, that the pharmacokinetics, ADME. So today it is the drug and target interaction, how to optimize the drug target interaction for having the maximum efficacy, that we are going to learn today. Okay, so like how we do it, so like every each and every session, like we are used to doing the same. So there are 20 questions today. All questions are the MCQ questions. Okay, and each and every question will have 4 different answers. What you have to do after showing a particular question, I will wait for some time, and you have to let me know in the chat box A, B, C or D which of the following will be the correct answer for that particular question. Okay, and after some time I will check the chat box again, and I will check your answers, and then I will show you the particular correct answer, and we will discuss that particular question in detail. Okay, the reason why we do it, that in the assignments, the weekly assignments, what you have, what you are doing are the MCQ in pattern, right? So because of that, we do this MCQ kind of question so that you can easily solve your weekly assignments. Okay, and also each and every question we will followed up with a descriptive discussion regarding that particular topic. So it will help you to answer the subjective questions which may be asked in your final examinations. Okay, and also it will let you understand the course and that particular topic better. So I hope there is no doubt or query from our previous class. If there is, please let me know. We can discuss it today or in the next day. I will surely discuss that particular thing. Okay. So with that, I will like to start the session with the first question of today's class. Okay. Just let me show you the first question. So the first question, it says that which of the following statements best describes structure-activity relationship or SAR? Your options are number A, the study of which functional groups are important to the chemical reactivity of the drug. Number B, the study of the physicochemical properties that are important to the absorption of the drug into the blood supply. Number C, the study of the structural features of the drug that are important to its biological activity. And number D, the study of the structural features of a drug that are important to its chemical stability. Which of the following statements best describes the structure-activity relationship? Again, I will repeat number A, the study of which functional groups are important to the chemical reactivity of the drug. Number B, the study of which physicochemical properties that are important to the absorption of the drug into the blood supply. Number C, the study of the structural features of the drug that are important to its biological activity. And number D, the study of the structural features of the drug that is important to its chemical stability. Which of the following statement best describes the term structure-activity relationship or SAR? Let me wait for a minute and two and then I will check the chat box. Okay. A, B, C or D, whatever you think is the correct answer, please let me know. Read all the options carefully and then try to answer the question. Okay. I will wait for a minute or two, then I will check the chat box. A, B, C or D, let me know which of the following you think is the correct answer for problem number 1. The problem number 1, again I repeat, which of the following statements best describes the structure-activity relationship? Your options are number A, the study of which functional groups are important to the chemical reactivity of the drug. Number B, the study of the physicochemical properties that are important to the absorption of the drug into the blood supply. Number C, the study of the structural features of a drug that are important to its biological activity. And number D, the study of the structural features of a drug that are important to its chemical stability. Which will be the correct answer for problem number 1? Which of the following best describes the SAR or structure activity relationship? Let me check the chat box. I hope there will be some answers. Okay, I got only one answer which is saying that number A is the correct one. Others have not answered. Okay, let me check if number A is the correct answer for problem number 1. So the correct answer will be, it is actually number C. Okay, the study of the structural features of the drug that are important to its biological activity. It is known as the structural activity relationship or SAR. Now let us understand what is the SAR or structure activity relationship. So basically, once the structure of the lead compound is known, the medicinal chemist moves to the study of the structural activity relationship. And what is the lead compound? That we have already discussed in our previous classes couple of times. Okay, so I am not going to elaborate on that part. So the aim is to identify those parts of the molecule that are important to the biological activity and those that are not. If it is possible to crystallize the target with the lead compound bound to the binding site, the crystal structure of the complex could be solved by extracrystallography, then studied with the molecular modeling software to identify the important binding interactions. However, it is not possible if the target structure has not been identified or cannot be crystallized. It is then necessary to revert the traditional method of synthesizing selected number of compounds that vary slightly from the original structure, then studying what effect that has on the biological activity. One can imagine the drug as a chemical knight entering into the body in order to make a battle with an affiliation. The drug is armed with variety of weapons and armor, but it may not be obvious which weapons are important to the drug's activity or which armor is essential to its survival. We can only find this out by removing some weapons and armor to see if this drug is still effective. The weapons and armor involved are the various structural features of the drug that can either act as a binding group to the target's binding site or assist and protect the drug on its journey to the body. Recognizing the functional group and the sort of the intermolecular bonding bonds that can form the important and is understanding the how the drug might bind to its target. Okay. So, in a nutshell, the structure the activity relationship is the structure that determines the function. It is presented by the Crum and Brown and Fresher in 1865. The structure of the compound determines the biological effect it has. Determination of the structural function relationship helps the medicinal chemist to synthesize new drugs and pharmaceutical agent. For example, this is a drug. Okay, and here are a couple of functional groups that are involved. So here, let's see. So there is a three rings are there, number A, number B, and number C. So on the number A, there are three methoxy groups. Okay, so this methoxy group is important for the correct conformation and activity. This methoxy group is not required or it is not essential. Okay, and this methoxy is also important for binding ability. So if you change this methoxy group or you can remove this methoxy group, it won't have much activity or it won't have much effect on this drug's activity. This particular bond unsaturation in this position increases this drug's activity. Ring expansion is not permitted. Okay, so this B ring cannot be expanded beyond this point. Epimer, so in this position, epimer diminishes the activity. This NHCOMe group can be replaced by other substituent. Okay, this number C allows the ring contraction. So the C ring can be contracted. It won't have any effect on the drug's ability. This OMe group permits halides, alkyl, alkoxy, amino group, but not the bulky groups. Okay, so here bulky groups are not permitted. So these are the example of the structure activity relationship. How a particular feature of a drug or structure is important to its activity, and this is known as the structure activity relationship. Okay, so this option number C, which says that the study of the structural features of the drug that are important to its biological activity, it is known as the structure activity relationship. Okay, now let me go to the next problem. Problem number 2, it says that SAR shows that the important binding groups in morpholine are the phenol, aromatic ring, and amine. A diagram can be drawn which shows these functional groups and the relative orientation. What is the term for this diagram? Your options are number A, pharmacodynamics, number B, pharmacokinetics, number C, chromophore, and number D, pharmacophore. Again, I will repeat. SAR shows that important binding groups in morpholine are phenol, aromatic ring, and amine. A diagram can be drawn which shows these functional groups and their relative orientations. What is the term for this diagram? Your options are number A, pharmacodynamics, number B, pharmacokinetics, number C, chromophore, and number D, pharmacophore. Again, let me wait for a minute or two, then I will check the chat box. Okay, let me know A, B, C, or D, which of the following you think is the correct answer for problem number 2. What is the term used for this particular diagram? Let me know A, B, C, or D. Which of the following you think is the correct answer? Even if your answer is wrong, it's fine, but try to answer the question. Okay, let me check the chat box. Let's see if there are any answers for our problem number 2. Okay. I have received only one answer, which is saying it that number C is the correct one. Let me check if number C is the correct one or not. So the correct answer will be, it is number D. Pharmacophore is the term which is used to describe the diagram that can be drawn to show the functional groups and their relative orientation in a particular drug. Okay. So what is a pharmacophore? Now let us understand this part. So a pharmacophore is an abstract representation of the essential chemical features and their spatial arrangement required for a molecule to be recognized by a biological target like a receptor or enzyme and trigger a biological response. According to IUPAC, it is an ensemble of the steric and electronic features that is necessary to ensure the optimal supramolecular interaction with a specific biological target. Key components of the pharmacophoric model: instead of specifying actual atoms, a pharmacophore uses generalized features when represented as the spheres, vectors, and plane. Hydrogen bond donor and acceptor: features that can participate in hydrogen bonding. Hydrophobic center: regions that engage in lipophilic interactions such as alkyl chain, aromatic rings essential for pi-pi stacking or cation-pi interaction. Positive and negative ionizable group: basic or acidic group that form the ionic bonds. Exclusive volumes: forbidden areas that represent the physical boundaries of the binding site and used to prevent steric clashes. Common pharmacophoric features: hydrogen bond acceptor, atom with a lone pair to accept the hydrogen bond. Common structural examples: amine, ketone, carboxylate. Hydrogen bond donor, atom with a hydrogen to donate. Alcohol, amide, amides, amines. Aromatic ring, rings that capable of pi-pi stacking like phenyl ring, indole. Hydrophobic interaction, non-polar region like alkyl group, halogen substituent. And ionizable, positively negatively charged group like ammonium ion, carboxylate ion, like with the positive and negative ions. Okay, so these are the common pharmacophoric feature types. Okay, so remember this particular group, which is basically which describes the relative orientation of the functional groups and their biological activity, is known as the pharmacophore, and these are the common features of the pharmacophoric model. There is something also which is known as a 3D pharmacophore, which we will discuss in our upcoming slides and in this class itself. Okay, so this is all about the pharmacophore. So is it clear, or is there any doubt what is a pharmacophore and what is its role? If there is any doubt, please let me know. I want all of you to answer in the class. Okay, even if your answer is wrong, it's perfectly fine. Okay, there is no problem even if your answer is wrong. But please try to answer the questions. Otherwise, I won't understand if you are following the class or not. Right? Okay. So let me now go to the next problem. So problem number 3 says, Combristatin is an anti-cancer agent discovered from an African plant. Analog 1, this combristatin is the structure of combristatin, okay? So analog 1 is more active than combristatin, whereas analog 2 is less active. Which strategy is used in designing these analogs 1 and 2? Again, I will repeat, combristatin is an anti-cancer agent discovered from African plant. Analog 1, this is the structure of combristatin. Analog 1 is more active than the combristatin, whereas the analog 2 is less active. Which strategy is used to design these analogs 1 and 2? Your options are: number A, simplification; number B, isosteric replacement; number C, rigidification; and number D, chain contraction. Which of the following strategies is used to design these analog 1 and 2 of the combristatin? Simplification, isosteric replacement, rigidification, or chain contraction. Again, I will wait for a minute or two and then I will check the chat box. Let me know A, B, C, or D, which of the following is the correct answer for problem number 3. Which strategy is used to design the analog 1 and 2 of combristatin? Which of the following will be correct answer? Let me know. I will wait for a minute and then I will check the chat box, okay? Okay, let me check if there is any answer in the chat box. Again, I have received only one answer, which is saying that number C is the correct one. Okay. Okay, let's check if number C is the correct answer or not. So the correct answer is yes, it is number C. Rigidification is the method or the strategy which is used to design the analog 1 and 2 of chromostatin. So what is rigidification of the structure and how it works? Let us understand that. So, rigidification of the structure has often been used to increase the activity of the drug or to reduce the side effect. In order to understand why this tactic can work, let us consider let us consider a hypothetical neurotransmitter. This one, let us consider that. Okay. This is the quite simple flexible molecules with the several rotable bonds. Okay, I think yeah. Flexible molecules with several rotable bond can lead to large number of conformation or shapes. One of these conformation can be recognized by the receptor and is known as the active conformation. The other conformation are unable to interact efficiently with the receptors and are inactive conformation. However, it is possible to different receptor exists there which is capable of binding to one of these alternative conformation. If that is the case, then our model neurotransmitter could switch to two different receptors and give two different biological response, one of which is desired, one of which is not. The strategy of the rigidification is to make the molecule more rigid such that the active conformation are retained and the number of other possible conformation is decreased. This should reduce the possibility of the other receptor interactions and the side effect. The same strategy should also increase the activity. By making the drug more rigid, it is more likely to be in active conformation. When it approaches to the target binding site, it should bind more readily. This is an important when it comes to thermodynamics of binding. A flexible molecule that has to adopt a single active conformation in order to bind the target, which means that it has become more ordered. This results in decrease in entropy and the free energy of binding related to the entropy of the equation delta G equal to delta H minus T delta S. Any decrease of the entropy will adversely affect the delta G. In turn, it lowers the binding affinity which is related to delta G by equation delta G equal to minus RT ln K1. The total rigid molecule, however, is already in its active conformation and there is no loss of entropy involved in binding to the target. If the binding interactions is exactly the same as for the flexible molecule, the rigid molecule will have a better overall affinity. Okay, so this is an example. Like this is a flexible messenger to in the active conformation and these bonds are rotable bonds and this is a flexible conformation of the inactiveness. So for example, this is a active conformation, it is the inactive conformations, but both are the same conformation of a molecule. But if we can rigidify these flexible bonds into like this kind of ring system, so what will happen, the molecule will be rigid and it will require less energy to bind or to the receptor, means it will more readily bind to the receptor. Same happens with the combristatin. As you can see in combristatin, this part it is quite flexible, right? So these are the rotable bonds and this can rotate freely. But once you introduce a double bond, there will be only two conformations possible. One is number A and number 2. And according to our hypothesis, number A is the more active conformation, and rather than number B, you can also rigidify this analog 1 if you make a particular it a part of a ring. So for suppose if you can make this double bond a part of a ring like this, the compound it will be more active. So because then this trans conformation won't be possible and there will be only one conformation and that is analog one which has the better affinity. So this is the example of rigidification and how the rigidification occurs. Okay, now let me go to the next problem. So problem number 4, it says that which of the following is the or are the is a major aim in the drug design? Which of the following major aims in drug design is not related to the pharmacodynamics of the drug? Which of the following major aims of drug design is not related to the pharmacodynamics of the drug? Your options are: number A, the reduction of the side effect; number B, the maximization of activity; number C, the reduction of toxicity; number D, the maximization of oral bioavailability. Which of the following major aims of the drug is not related to the thermodynamics? Pharmacodynamics. If your options are: number A, the reduction of side effect; number B, the maximization of activity; number C, the reduction of toxicity; and number D, the maximization of oral bioavailability. Again, I will wait for a minute and then I will check the chat box. Let me know A, B, C, or D, which of the following you think is the correct answer. Try to answer every question. Even if your answer is wrong, it's fine, but at least try. Okay, let me check the chat box. Let's see if there is any answer for problem number 4. Okay, number D and number A. Let's see which of the following will be the correct answer. So the correct answer will be, it is number D, the maximization of the oral bioavailability is not related to the pharmacodynamics of a particular drug. Okay, so now let us understand what is the pharmacokinetics that we have discussed in number of time till now. So the drug is administered, it is absorbed, and then we have the distribution, metabolism, and elimination. Okay, so it is the ADME: absorption, distribution, metabolism, and elimination or excretion. Okay, so what is the drug distribution? So after that, this is till the after so till now we are learning what is our body is doing to a drug. Then comes to the action of the drug, which is the transfer. So first is the after the distribution, the transfer to the site of action. The regulatory proteins are there like receptor, enzyme, ion channel, and transporter. Then binding to the molecular target, which is described by the drug's affinity. Then mechanism of drug action, and then response of the drug, which is known as the efficacy. So in pharmacodynamics, two things are very important. One is affinity and the another is efficacy. Okay, affinity is the is how strongly the drug is binding to its target, and efficacy how it is responding to a action of the drug. So the first three aims, one is the reduction of side effect, maximization of activity, and reduction of toxicity, all are related to the pharmacodynamics. But the maximization of oral viability, it is not related to the pharmacodynamics, rather it is related to the pharmacokinetics or the drug administration. That is related this aim is related to that part. Okay, so not the pharmacodynamics. So the primary aim of the pharmacodynamics is to determine what a drug does to the body by identifying its biochemical, physiologic, and molecular effect. Primary objective: identify the mechanism of action, determine how a drug interacts with the specific cellular target such as the receptors, enzyme, or ion channels to produce a biological response. Establish dose response relationship, quantify the relationship between the drug concentration and the intensity of the effect, determine the potency, the amount needed, or efficacy, the maximum effect. effect. Define the therapeutic window, identify the dosage range that provides the maximum benefit while minimizing adverse effects or toxicity. Assesses safety and toxicity, investigate the undesirable effects on key physiological functions such as the respiratory or cardiovascular system to ensure patient safety, and predict variability, understand how factors like age, genetics, disease alter the individual's response to the drug. So these are all the primary objectives of the our pharmacodynamics. Okay. So number D was the correct answer. Now let me go to the next problem. Okay, so there are some clinical and industrial applications also of pharmacodynamics. Why it is important? The drug development used in the preclinical and clinical trials to select the lead compounds, determine the first in human doses, and design optimal drug dosing regimen. Personalized medicine, tailoring drug therapies to an individual's unique genetic profile or physiological status to maximize the efficacy. Therapeutic drug monitoring, guiding the clinicians in adjusting the doses for the drugs with a narrow therapeutic index to keep the blood levels within a safe and effective range, managing drug interaction, understanding how multiple medication might interact at the same receptor site to enhance and reduce the overall effect. So these are the clinical and industrial applications of the pharmacodynamics. So I hope it is clear what is the pharmacodynamics and what are the aims and applications. If you have any doubt or any query, please let me know, okay? I will try to repeat that question even if there is any doubt or query. Let us go to the next problem. Problem number 5 says, which of the following strategies was used to develop anti-cancer drug crizotinib? Your options are number A, ring fusion, number B, ring cleavage, number C, chain contraction, and number D, all of the above. Which of the following strategies was used to develop anti-cancer drug crizotinib? Ring fusion, ring cleavage, chain contraction, or of the above? Again, I will wait for a minute, then I will check the chat box. Okay, let me know A, B, C, or D, which of the following you think is the correct answer for problem number 5. Which of the following strategies was used to develop anti-cancer drug crizotinib? Number A, ring fusion, number B, ring cleavage, number C, chain contraction, number D, all of the above. Which of the following strategies was used to develop anti-cancer drug crizotinib? Let me check the chat box. Okay. Both of the answers are saying that number D is the correct one. Let's find out if it is so. So the correct answer will be yes, it is number D. All of the above strategies was used to develop the anti-cancer drug crizotinib. So basically, this is the structure of crizotinib. As you can see, it may seem a little difficult, a little complicated, but it is not. Let us understand the thing. Okay, first. So this is an example of the structure-based drug design. Okay, so the structure-based drug design is normally used to observe the binding interaction of a ligand and then to identify the modification that will result in better interactions and greater activity. This approach was used to design of a recently approved anti-cancer drug called crizotinib and included a substantial modification which totally altered the scaffold of the molecule. PHA 665752. So this is PHA 665752. This molecule, this complete molecule, okay. So this was the starting point of this research and had been obtained from the structure-based drug design of the previous lead compound. However, it has a large molecular weight and too hydrophobic to be orally active. As you can see, this molecule It is so large, and there are very less hydrophilic groups, so it is very hydrophobic. And when the drug is very hydrophobic, it will not be orally active. Okay, so as we know, as the Ripilski's rule, that higher the molecular weight, the less orally active the drug will be. The structure was co-crystallized with the target enzyme, and the crucial binding interaction was identified. This included the dihydroindolin ring system, which formed two important hydrogen bond. Okay, so this ring system is important, that was found from the co-crystal structure with the target. This indolin ring is important because it has the hydrogen bond donor and hydrogen bond acceptor it together. Okay, so as well as the dichloroaromatic ring. So this dichloroaromatic ring, it was also important. So as a result of the study, it was noted that much of the scaffold connecting these binding groups was reductant, and so much simpler, less hydrophobic skeleton was designed, which would position the important binding groups in similar fashion but more efficient manner. The thought process behind the design involved a ring fusion, ring cleavage, and chain contraction. When the novel structure was synthesized, they were found to bind as predicted, and further structure-based design was used to optimize to process the leading compound, the crizotinib. So this group is important, this part is important. Other parts was not that much important. So first is the ring fusion. So this particular part, first it was fused. Okay, so so that there is a hydrogen bond donor acceptor is intact. Then the thought two was a ring cleavage. Then after the cleaving of the this ring, so the ring cleavage leads to this compound. If you cleave this part, it will lead to this compound where also hydrogen bond donor acceptant part was as it is. Then chain contraction. If you contract this particular chain part, so you will lead to the this. So this is the novel scaffold which bind it with the target exactly with this point. Okay, and then what we have this now optimization gives us this crizotinib. So after this is one example, and it is a very good example how the structure based drug design happen in today's research. Okay, so first remember is a ring fusion, ring cleavage, and chain contraction. Three different strategies was used to develop this crizotinib from the starting point PHA 65752. Okay, just you have to keep the parts that are interacting unchanged, and then you can modify the other part, but you have to modify it in a way that the previous interaction, they should be there in the new structure also, right? Now let me go to the next problem. Problem number 6 says succinylproline was the lead compound for captopril, which acts as an inhibitor of the angiotensin converting enzyme. What is the relevance of the thiol group in captopril? Your options are: number A, it can form covalent disulfide bond with cysteine thiol groups in the active site. Number B, it acts as an isoster of carboxylic acid. Number C, it acts as a bioisoster of the carboxylic acid. Number D, it is a smaller group than carboxylic acid and fits the binding site easily. What is the role of the thiol group in the lead compound captopril? Okay. Number A, it can form a covalent disulfide bond with the cysteine thiol groups in the active site. Number B, it acts as an isoster of carboxylic acid. Number C, it acts as a bioisoster of carboxylic acid. And number D, it is a smaller group than the carboxylic acid and fits the binding site more easily. Let me know A, B, C or D, which of the following you think is the correct answer for problem number 6. I will check the chat box after a minute. Okay, let me know A, B, C or D, which of the following you think is the correct relevance of the thiol group in the captopril, in the development of captopril from the succinylproline. Okay, let me check if there is any answer in the chat box. Okay, number B, number C. So I have received two answers, number C and number B, two different options. Let me check number B or number C, which of the following will be the correct answer. So the correct answer will be, it is number C. It acts as a bioisoster of carboxylic acid. Okay. So there is a substantial difference between this isoster and bioisoster. Let us understand first what is the bioisoster. Okay. So in medicinal chemistry, a bioisoster is a chemical group or substituent that has similar physical or chemical properties to another group and produces broadly similar biological effects in a compound. Bioisosteric replacement is a core strategy in drug design used to optimize a lead compound's properties such as increasing potency, improving solubility or reducing toxicity without significantly altering its overall structure. Classification of bioisoster. Bioisosters are generally divided into two main categories. One is the classical bioisostar, another is the non-classical bioisostar. So what are the classical bioisostars? So let me just highlight that part for easier understanding. So generally two main categories of isostar is the one is the classical one and another is the non-classical bioisostar. Okay. So what is the classical bioisostar? These involve the substitution of atoms or group with similar electronic configuration, same number or valence electron, and steric properties, similar size and shape. For example, monovalent replacing hydrogen with a fluorine atom to block metabolic oxidation and extends drug half-life. Divalent replacing methylene group CH2 with an oxygen or sulfur atom. Ring equivalent swapping benzene ring with thiophene or pyridine ring. These are the example of the classical bioisostar. Now, what is a non-classical bioisostar? These do not strictly follow the electronic and steric definition of the classical isostar, but mimic the biological function of the original group similar binding interaction. For example, carboxylic acid mimic replacing carboxylic with the tetrazole ring to improve the bioavailability and metabolic stability, as seen in the antihypertensive drug losartan. Amide mimic swapping the amide group with the triazole or oxazole to increase the resistance to the enzymatic hydrolysis. Why use bioisostar? To enhance potency, fine tuning of how the drug binds to the target receptor, improve pharmacokinetics, increasing water solubility or membrane permeability of to ensure the drug reaches to the target effectively, reduce toxicity, replacing a group responsible for side effects or the safer alternative that retains the therapeutic activity, and circumvent patents. Discovering bioisostar of patented compounds can allow for the development of new independent intellectual property. So these are the reasons why bioisostars have been used in medicinal chemistry extensively. Okay. So now according to the question, so this is the succinyl proline which has the IC50 value of 628 micromolar. So the development of captopril from succinyl proline, as you can see, first there is an introduction of the CH3 group in this position. So the introduction of the CH with R conformation, it decreases the IC50 value to 52 micromolar, and then replacing only this carboxylic group with the thiol, it decreasing the increase IC50 value furthermore to the 23 nanomolar only. Okay. So the only two changes have been done on the succinyl proline to reach to the captopril. Okay. One is the introduction of methyl group and another is the replacement of the carboxylic acid to thiol. So here thiol is the bioisosteric replacement of the carboxylic acid group. So carboxylic acid group in the normal drug, it is not a very good to have a free carboxylic acid group in the particular drug due to the enzymatic cleavage and all these reasons. So what are the bioisostars of carboxylic acid that can be used? So basically very crucial and very actually important isostar is the tetrazole. Okay, and it is very inconvenient. So it is a example of the non-classical bioisostar. We have the hydroxychromine, also acyl sulfonamide, this one sulfonamide, sulfonic acid, phosphamide, oxothiazole, oxoxarazole, hydroxythiazole, hydroxyisoxazole, isothiazole. We have acyl cyanamide and then hydroxymic acid. So all these are the examples of the bioisosters of the carboxylic acid. Okay. So this was all about your problem number, I think it was the problem number 6. Now let me go to the next problem. So problem number 7 says the procedure of transforming racemic rings to a single enantiomer is called, your options are number A, chiral pool, number B, asymmetric synthesis, number C, chiral switch, number D, asymmetric switch. The procedure of transforming racemic drugs to a single enantiomer is called which of the following? Chiral pool, asymmetric synthesis, chiral switch, or asymmetric switch? Let me know A, B, C, or D, which of the following will be the correct answer for problem number 7. Let me wait for a minute. Then I will check the chat box. Let me check the chat box. Let's see if there is any answer. Okay, number B is the answer which I have received. Let me check if number B is the correct one or not. Okay, so the correct answer will be, it is number C, chiral switch. Okay. So what is a chiral switch? A chiral switch is the pharmaceutical development strategy where a drug previously marketed as racemate, a 50-50 mixture of two mirror image enantiomer, is redeveloped and re-released as a single purified enantiomer. The goal of this switch is typically to isolate the eutomer, the active enantiomer, and to remove the distomer, which is the less active or potentially toxic enantiomer, which is sometimes viewed as the isomeric ballast or an impurity. Core reasons of a chiral switch: enhanced efficacy and safety. Removing the distomer is that is inactive and responsible for side effect can improve the therapeutic index. Predictable pharmacokinetics. Single enantiomer often show less inter-individual variability in metabolism, as seen as the esomeprazole compared to the racemic omeprazole. Patent extension or evergreening. The companies often launch a single enantiomer version near the patent expiry of the original racemate to maintain the market exclusivity and the protect revenue from the generic competition. The common drug examples, so for example, the racemic precursor omeprazole, chiral switch, or the single enantiomer is esomeprazole. The common brand name is the Nexium, and claimed benefit: better acid control, more consistent metabolism. Citalopram, escitalopram is the chiral switch, which is known as the Lexapro, the higher potency, faster onset of action. Albuterol is a levabuterol, which is the Zopenex, which is the claimed benefit is increased potency and potentially fewer side effect. Ofloxacin, levofloxacin, which is a higher antibacterial property. The cetirizine or levocetirizine, which is the Zizal, which is increased potency, claimed the lower sedation. So these are the examples of the chiral switch. So notable successes and failures: the thalidomide lesson. Thalidomide is the classic warning of chirality. The R enantiomer is sedative and S isomer was teratogenic, which causing the birth defect. However, the poor candidate for the chiral switch because it undergoes rapid in vivo racemization, meaning the pure enantiomer convert back to each other inside the body. Aborted switches. Some attempts failed during the development due to the unexpected toxicity. For example, R-fluoxetine, the switch for the Prozac, was halted after the clinical trials showed it caused abnormal heart rhythm. Okay. So this is the examples and reasons of development of the chiral switch. So now I will go to the next problem. Problem number 8. It says the use of one drug for activating different drugs is called dash. Your options are: number A, additive therapy; number B, combinatorial therapy; number C, MT or multiple target therapy; and number D, none of the above. The use of one drug for activating different targets is called which of the following? Number A, additive therapy. Number B, combinatorial therapy. Number C, empty therapy or multiple target therapy. And number D, none of the above. Let me know A, B, C or D, which of the following is the correct answer. Again, I will wait for a minute, then I will check the chat box. The use of one drug for activating different targets is called which of the following? Let me check the chat box. Okay, I have received one answer which is saying number C is the correct one. Let's see if it is the correct answer or not. So the use of one drug for activating different targets is called, yes, it is no, number C, which is the multiple target therapy. So what is the multiple target therapy? So designing the drug that interact with more than one target. So many diseases require a cocktail of drugs interacting with different targets to provide suitable treatments. A better approach would be to design the agents that would interact with two or more targets in a controlled fashion in order to reduce the number of drugs that have been taken. This is known as the multi-target drug therapy or MTDD. There have been two approaches to designing such multi-target directed ligands. One is to design the agent from the known drugs and pharmacophores such that the new agent has combined properties of the drug involved. Other approach is to start from the lead compound which has activity against the wide range of the targets that and then modify the structure to try and narrow the activity down to the desired targets. So one drug that is going to interact with two targets and both have a synergetic effect which is known as the multiple target. So what are the advantages of this multiple target therapy? Higher efficacy against complex diseases such as cancer, more predictable and superior pharmacokinetic and safety profile as a consequence of administrating a single drug. The combination of multiple active principles may result in positive but also negative synergetic effect which limits the number of the useful combination. The probability of developing target-based resistance simultaneously to multi-target drugs is statistically lower than the probability of developing resistance against the single target drug. The risk of the drug-drug interaction associated with the combination therapies is avoided and the therapeutic regimen is greatly simplified. The use of the drug cocktails often complicates dosing schedule and negatively impacts the patent complication. It's more even difficult to perform the clinical trials to drug combination if the drugs come from different companies. Okay, so all these are the advantages of the multiple target therapy. So if you are, for example, you are given to write a short note on it, you have to first write what is the multiple target therapy. Preferably, you have to draw this kind of figure, and then you have to note down the advantages of this kind of therapy. Okay. So this is all about the multiple target therapy. Now I will go to the next problem. So problem number 9 says, by which method chimeric drugs are formed? Your options are number A, in silico drug designing method, number B, pharmacophoric hybridization, number C, bioisosteric replacement, and number D, structure based drug design. By which of the following method chimeric drugs are formed? Number A, in silico drug designing method. Number B, pharmacophoric hybridization. Number C, bioisosteric replacement. And number D, structure based drug design. Let me know A, B, C or D, which of the following will be the correct answer for problem number 9. By which method chimeric drugs are formed? By which method chimeric drugs are formed? In silico drug designing method, pharmacophoric hybridization, bioisosteretic replacement, or structure-based drug design. Let me check the chat box. Let's see if there is any answer for problem number 9. Okay, so I think I have received two answers. One is saying number A and another is saying number B. So let's find out number A or number B, which will be the correct answer. So the method by which the chimeric drugs can be formed is, it is number B, pharmacophoric hybridization. So what is chimeric drugs? Another method is to design a chimeric drug that contains a key pharmacophoric features of two drugs. So basically chimeric drugs are the drugs that contain the main pharmacophoric features or the main important active features of two different drugs in a single molecule. Okay. For example, here you can see there one drug is, this is the colchicine. Okay. Another drug we have this 2-methoxyestradiol. Okay. So these both are actually anti-cancer drugs. So one is the colchicine and then 2-methoxyestradiol. Now, when we take the pharmacophoric features of this two drug, for example, this trimethoxy group and this group and this particular group from this side. So the blue one, this blue one from this drug and this blue part from this drug, and then combine the two in a single drug. So we find out a chimeric drug. Okay. So the structure containing features of true-methoxyestradiol and colchicine has been synthesized as a potential anti-cancer agent. Although both of the parent structure have anti-cancer activity, they have serious drawbacks. 2-methoxyestradiol is metabolized rapidly, while colchicine has the toxic side effect. The chimeric structure, this has the anti-cancer property, but with improved pharmacokinetic properties and less toxicity. Okay, so that is why development of chimeric drugs are very important. Okay, because it's to it for the improved efficacy and lesser toxicity. And one very important method for developing the chimeric drug is the pharmacophoric hybridization method. Now, what is the pharmacophoric hybridization? So, pharmacophoric hybridization, often used interchangeably with the molecular hybridization, is a drug design strategy that involves covalently combining two or more essential chemical fragments or pharmacophores from different bioactive molecules into a single chemical entity. This approach aims to create multi-target drugs that offer improved efficacy, reduced toxicity, and a lower risk for the drug resistant compared to the individual parent compounds. Okay. So this is the pharmacophoric hybridization. So chimeric drugs, remember, is a drug that contain two or more pharmacophoric features of the parent drugs of to different drugs combined to a particular drug. Okay, so this is known as the pharmacochimeric drug, and the method is known as the pharmacophoric hybridization. Okay. Now let me go to the next problem. So problem number 10 says compound number 2 is more interactive with the target rather than the parent lead compound 1. What is the reason? Your options are number A, the strategy that was used for one of the ring expansion. Number B, blue nitrogen allows better solubility. Number C, blue nitrogen can act as a hydrogen bond donor. Number D, the strategy used could be described as the extension since further binding interaction has occurred. Which of the following is the reason for this compound 2 to be more active than lead compound 1? Your options are number A, the strategy used was one of the ring expansion. Number B, the blue nitrogen allows better solubility. Number C, blue nitrogen can act as an hydrogen bond donor. And number D, the strategy used could be described as the extension since a further binding interaction has occurred. Let me know A, B, C or D, which of the following will be the correct answer for problem number 10. Compound number 2 was found to be more interactive with target rather than the parent compound 1. What can be the following reason? Number A, the strategy used was one of the ring expansion. Number B, blue nitrogen allows better solubility. Number C, the blue nitrogen can act as an hydrogen bond donor. Number D, the strategy used could be described as extension since one binding interaction has occurred. Let me check the chat box. Let's see what are the answers that I have received. Okay. So number D is given as the answer. Let us find out if number D is the correct answer or not. So the correct answer will be yes, it is number D. The strategy used could be described as an extension since the further binding interaction has occurred. So now what is the extension of the structure? The strategy of extension involves the addition of another functional group or substituent to the lead compound in order to probe for extra binding interaction with the target. Lead compounds are capable of fitting the binding site and have the necessary functional groups to interact with the sum of the important binding regions present. However, it is possible that they do not interact with all the binding regions available. For example, a lead compound may bind to the three binding regions in the binding site but fail to use the fourth. For example, there is a drug in the binding site and the drug have some functional group that can interact with the three different binding region, but there may be another or one or two binding region that are unused by the particular drug. So what we have to do to use this extra binding region? So we have to use this extension strategy. What is this extension strategy? Extension tactics are often used to find extra hydrophobic region of the binding site to addition of the various alkyl or aryl alkyl group. These groups can be added to first functional groups such as alcohol, phenol, amine, carboxylic acid that should be present in the drug as long as it does not disrupt the important binding interaction that are already present. Alternatively, they could build into the binding building blocks used to synthesize of the various analog by some token substituents containing polar functional groups could be added to probe for extra hydrogen bonding or ionic interaction. So here this particular nitrogen is added. So after adding this nitrogen, one more binding interaction has occurred. Okay, so that is why this drug number 2 is more active than drug number 1 and it is known as the extension strategy. Okay. Now I will go to the next problem. Problem number 11 says Du-122-290 is more potent and selective than saltopride. What is the reason? Your options are number A, spiral ring has been made it more rigid. Number B, replacing amide moiety has improved the lipophilicity. Number C, both A and B are correct. Number D, it cannot be said surely from the structure. So this is the structure of saltopride and this is the structure of Du-122-290. Okay. So this Du-122-290 is more potent than this parent drug saltopride. What is the reason? Now, what is the reason? Your options are number A, pyrrole ring has made it more rigid. Number B, replacing amide moiety has improved lipophilicity. Number C, both of the A and B are correct. And number D, it cannot be said for sure only from the structures of the compounds. Let me know A, B, C or D which of the following will be the correct answer for problem number 11. I will wait for 2 minutes and then I will check the chat box. Okay. What is the reason why behind the more potency and selectivity of Du-122-290 than saltopride? Let me check the chat box. Let's see if there is any answer. Okay. Okay, I think there is no answer for that problem number 11. Let me show the correct answer and let us discuss it. So, the correct answer will be it is number A. Pyrrole ring has made this compound more rigid. Okay. So the pyrrole ring, so here as you can see in sultopride, there is this amide linkage. Okay. And in case of this Du 122290, there is a pyrrole ring that has replaced this amide linkage with this particular ring. Okay. So the pyrrole ring of the Du 122290 serves to increase the rigidity of the side chain present in the sultopride. So here replacing this amide linkage with this has has decreased the number of rotable bonds, thus increase the rigidity. The red colored bond of the sultopride, this one is freely rotable but is locked when the pyrrole ring in the as the pyrrole ring in the Du 12290. This reduces the number of possible conformations that this Du 122290 can adopt and increases the chance of the active conformation being present when it enters the binding site, thus increasing the activity. Moreover, some of the conformation that are now disallowed may be those which were adopted when the sultopride binds to the D2 receptor, thus increasing selectivity for the D3 receptor. Okay, so this is the sultopride introducing the pyrrole ring as the bioisostar. So here one more thing that is very important to understand, that this particular pyrrole ring is a bioisostar of our amide linkage. Okay, so the introduction of this pyrrole ring as a bioisostar of amide linkage has improved the efficacy because it has introduced the rigidity, decreasing the number of active conformation and increasing the chance of this compound to binding more rapidly with the particular target with the better efficacy, thus increasing the selectivity also. Okay. Now I will go to the next problem. Problem number 12 says, which element act as a bioisostar of hydrogen? Your options are number A, D means deuterium, number B, F means fluorine, number C, lithium, and number D, both A and B. Which of the following element acts as a bioisostar of hydrogen? Deuterium, fluorine, lithium, and number D is the both A and B. Let me know A, B, C or D, which of the following will be the correct answer for problem number 12. Which element acts as a bioisostar of hydrogen? Deuterium, fluorine, lithium, or both A and B? Okay, let me check the chat box now. Okay, still there is no answer. Let me check the chat answer first. Okay, so the element that acts as the bioisostar of hydrogen will be, the correct answer is number D. Both A and B means both deuterium and fluorine can act as a bioisostar of hydrogen. Now let us understand the bioisostar of hydrogen and this thing. So, okay, sorry. Yeah. So in medicinal chemistry, a bioisoster of hydrogen is an atom or group that used to replace hydrogen in a molecule to improve its drug-like properties while maintaining its biological target interaction. Most common bioisoster of hydrogens are fluorine and deuterium. Okay, let me highlight this too. So the most common bioisoster of hydrogen are this fluorine and deuterium. Okay, so why fluorine and deuterium are the most common and most used bioisoster of hydrogen? Because hydrogen is the smallest atom and its bioisoster must be small to avoid disrupting the molecules fit into the target binding pocket. Fluorine is the most widely used bioisoster of hydrogen. Why it is used? Its size is very similar to hydrogen. The van der Waals radius is 1.35 Armstrong versus 1.20 of hydrogen, so it fits perfectly into the same spaces. Effect, it is highly electronegative and creates a very strong carbon fluorine bond that is resistant to metabolic oxidation by liver enzyme, the cytochrome P, and this can significantly extend the drug's half-life. Example, ezetimibe cholesterol lowering uses the fluorine to block the metabolic soft spots or the benzene ring. Okay, so this is the example. The ezetimibe, where fluorine is used as a bioisoster of hydrogen here in this place and in this place. So it is used to block the metabolic soft spots on the benzene ring. So in case of benzene ring, the para positions are basically very much more metabolically labile or soft spots. Okay, so metabolic degradation can happen in this and this spot when hydrogen is present. So, so that it does not happen, fluorine can be added as the isoster of the hydrogens to block the metabolic soft spots. Okay, and deuterium is stable, non-radioactive isotope of hydrogen. Why it is used? It is the perfect bioisoster because it is chemically nearly identical to hydrogen but twice as heavy. Effect, the CD bond is stronger and vibrates more slowly than a CH bond, leading to a deuterium kinetic isotope effect that slows down the metabolic breakdown. Example, deutetrabenzine was the first FDA approved deuterated drug requiring lower and less frequent doses than the non-deuterated counterpart. So this is the example of the deutetrabenazine, which is the first FDA approved deuterated drug. This is important to remember. Okay, that this drug is the first FDA approved deuterated drug requiring the lower and less frequent doses than its non-deuterated counterpart containing hydrogen. Okay, so these two drugs are very good examples where the fluorine and deuteriums are used as the bioisoster of hydrogen to improve the compound's efficacy and potency. Okay, so now I will go to the next problem. So problem number 13 says, in which of the following ways can 3D pharmacophores of a flexible structures be achieved? Your options are: number A, rigid analogues of the flexible compound could be synthesized and tested to see where activity is retained, where activity is retained. Number B, crystallize the target with the compound bound to the binding site. Number C, NMR spectroscopy to solve the active conformation of isotopically labeled molecules bound to their active site. And number D, all of the above. In which of the following ways can 3D pharmacophores of a flexible structures be achieved? Your options are: number A, rigid analogues of the flexible compound could be synthesized and tested to see where the activity is retained. Number B, crystallize the target with the compound bound to the binding site. Number C, NMR spectroscopy to solve the active conformation of isotopically labeled molecules bound to their active site. And number D, all of the above. Which of the following will be the correct answer for problem number 13? Let me know. Which is the correct way of achieving 3D pharmacophore of the flexible structures of a molecule? Let me wait for a minute, then I will check the chat box. Okay. In which of the following ways 3D pharmacophores of the flexible structures can be achieved? Let me check the chat box. Let's see if there is any answer in the chat box regarding the problem number 13. Okay. Two answers I have received, and both are saying that number D is the correct answer. Let's find out. So the correct answer will be yes, it is number D. All of the above are the ways in which the 3D pharmacophore of the flexible structures can be achieved. So, a 3D pharmacophore is a spatial model that defines the essential three-dimensional arrangement of the chemical features, such as atoms or functional groups, required for a drug molecule to bind to a specific biological target and trigger a response. Unlike a 2D pharmacophore, which focuses on connectivity and functional groups, a 3D model specifies spacious geometric constraints like distance, angle, and vectors. Core features and component: the pharmacophores abstract molecular structures into the features represented as geometric entities like sphere, vectors, and plane. Chemical features: common types include the hydrogen bond donor acceptors, aromatic ring, hydrophobic areas, and positively negatively ionizable group. Geometric constraint defines the distance between these features, and occasionally they are directionality, for example specific angle of the hydrogen bond. Exclusion volume: forbidden zone that represent the physical boundaries of the binding pocket where the atoms cannot reside due to the steric clashes with the target. Elucidation approaches: the structure-based approach, input data, 3D structure of the target like protein and ligand. Use case: based when the receptor's geometry is known. Ligand-based: a set of active molecules are the input data, and use case used when the target structure is unknown. Apo-based: empty target binding site, the de novo drug design without the known ligands. So applications in drug discovery: virtual screening efficiently filters libraries containing millions of molecules to identify the hits to match the required 3D pattern. Scaffold hopping: identify chemically diverse molecules that share the same spatial arrangement or features, allowing for novel drug structure. Lead optimization helps determine which parts of the molecule are critical for activity and which can be modified to improve potency or safety. Mechanism elucidation provides insight into how ligands interact with the receptors on the molecular and mechanistic level. So number of ways that can be achieved is: the rigid analogs of the flexible compound could be synthesized and tested to see whether the activity is retained. Crystallize the structure with the compound bound to the binding site, and NMR spectroscopy to solve the active conformation of the isotopically labeled molecules bound to their binding site. So all these are the ways by which this particular 3D pharmacophores of the flexible structure can be achieved. Okay. So this is all about the 3D pharmacophores, its application, core features, and also the number of ways that it can be achieved. Now let me go to the next problem. So problem number 14 says, which of the following is correct answer? So here you have this amine, then followed by this VOC chloride, you have this the A, and then CH3COCl, you have this B. Now you have to identify what is A and what is B. Your options are: number A is this one, NRR with COCH3, number B is the NRR with COCH3, and A is the NHR, and number B, N is this one. Which of the following will be the correct answer? Which of the following options are correct? Let me know. In this particular transformation, which of the following answer will be the correct one? Which of the following is the correct answer? Let me know, A, B, C, or D. Let me check the chat box now. Okay. Hmm. Number B is the only answer that I have received, the only option that I have received as answer. Let me check if it is correct or not. So the correct answer will be yes, it is number B. So, yeah. So basically, the binding role of amide. So first, let us understand. So this is the conversion is the dealkylation of the tertiary amine and formation of the secondary amides. Okay. So first, for the this reagent is used for the dealkylation, means to convert the NMe to NH. Okay, and then NaCH3O COCl makes it the secondary amide. So we have this tertiary amine to secondary amide conversion with this. So number B, which says that B is NH NRR, NR2, COCH3, it is the correct answer. Okay. So now let us understand the why amides are very important and what are the possible binding groups for the amide. So as you can see, when there is this is the primary amide. In case of this primary amide, this is the carboxylic, this carbonyl oxygen, it acts as a hydrogen bond acceptor because of this two lone pair which is here, and the two hydrogen on the nitrogen, it acts as a hydrogen bond donor. So in case the primary amide, we have two hydrogen donor and two hydrogen bond acceptor and two hydrogen bond donor interaction. Where in case of the secondary amide, one hydrogen is replaced by a alkyl group or R, and then we have this only one hydrogen bond donor but two hydrogen bond acceptor. In the tertiary amide, what happens? We do not have any hydrogen on this nitrogen, so there is no hydrogen bond donor, but the hydrogen bond acceptors are intact. Okay. So many of the lead compound currently studies in the medicinal chemistry are the peptides and polypeptides consisting of the amino acids linked together by the peptide or amide bond. Amides are likely to interact with the binding sites through the hydrogen bonding, and the carbonyl oxygen atom can act as a hydrogen bond acceptor and has the potential to form two hydrogen bond. Both of the lone pairs involved are the sp2 hybridized orbital, which are located in the same plane as the amide group. The nitrogen cannot act as a hydrogen bond acceptor because the lone pair interacts with the neighboring carbonyl group as I described earlier. Primary and secondary amides have the NH group, which allows the possibility of this group acting as a hydrogen bond donor. Okay. So this is what I have just already told. This is written in this position. So why amide linkage are very important? Because as you remember, when we were discussing about the protein, the peptide, primary, secondary, or tertiary, all the proteins, were the main peptide bond or peptide linkage or the backbone is basically a amide linkage. Okay. That is why this kind of interaction understanding is very important. Now I will go to the next problem. Problem number 15 says the enzyme responsible for the conversion of estrone to the female steroid hormone estradiol is, your options are number A NADH, number B beta hydrogenase, number C beta hydroxysteroid dehydrogenase, and number D alpha hydroxysteroid dehydrogenase. Which of the following enzyme is responsible for conversion of estrone to estradiol? Number A NADH, number B hydrogenase, number C hydroxysteroid dehydrogenase, and number D alpha hydroxysteroid dehydrogenase. Which of the following enzyme is responsible for the conversion of estrone to estradiol? Let me know. A, B, C, or D. Which of the following will be the correct answer? Let me check if there is any answer for problem number 15. Okay, number C is the option that I have received as answer. Let me find out if it is the correct answer or not. So the correct answer will be yes, it is number C. Beta hydroxysteroid dehydrogenase is the enzyme which is responsible for the conversion of estrone to estradiol. So this is the structure of estrone. As you can see here, we have a ketone group in the 17 position, and in case of estradiol, this ketone is converted to the hydroxyl group. So it is basically the reduction. Okay. So the enzyme catalyzed conversion of estrone to estradiol. So the enzyme 17 beta hydroxysteroid dehydrogenase type 1 catalyzes the conversion of estrone to the female steroid hormone estradiol, with the cofactor NADH acting as a reducing agent for the reaction. Inhibition of this enzyme may prove useful in treatment of estradiol-dependent tumors, as the level of estradiol present in the body would be lowered. The cofactor NADH is bound next to the estrone in the active site, and so it was a reason to the direct binding interaction between the estrone analog and NADH would lock the analog into the active site and block the access of the estrone itself. Therefore, the analog would act as an enzyme inhibitor. Various substituents were added in the position 16 to achieve this goal, as the crystallographic and molecular modeling studies had shown that the substituent would be ideally placed for an interaction with the cofactor. This led to the structure which shows the promising activity as an inhibitor. The amide group interacts with the primary amide NADH by hydrogen bonding, while pyridine ring interacts with the phosphate groups in the cofactor. A more conventional extension strategy was to add an ethyl group as C2, which allowed the additional van der Waals interactions with the small hydrophobic pocket in the active site that was absorbed to protons acted as the steric blockers and prevented NADH reducting the ketone group of the analog. Okay. So here the cofactor is NADH, okay, and this 17 carbonyl group is converted to hydroxyl with this enzyme 17 beta hydroxysteroid dehydrogenase type 1, which is also abbreviated as 17 beta HSD1, and the tumors or the cancerous cells which are dependent on the levels of estradiol, inhibiting this enzyme is maybe this enzyme is important or is critical for the treatment of that kind of cancers. Okay. Now I will go to the next problem. Problem number 16 says, which of the following statements is true regarding the isostar? Your options are number A. Isostars are the atoms or group of atoms which share the same valency but have different chemical or physical properties. Number B. Replacement of OCH2 with SCH2 retains the activity of the beta blocker propanolol. Number C. Replacement of OCH2 and NHCH2 eliminates the activity of beta blocker propanolol. Number D. Isosteric groups can be used to determine whether a group is involved in hydrogen binding or not. Which of the following statement is true regarding the isostars? Again, I will repeat. Number A, isostars are atoms or group of atoms which share the same valency but have different chemical or physical properties. Number B, replacement of OCH2 with SCH2 retains the activity of the beta blocker propanolol. Number C, replacement of the OCH2 with NHCH2 eliminates the activity of the beta blocker propanolol. And number D, isosteric groups can be used to determine whether a group is involved in hydrogen binding or not. Let me know A, B, C or D, which of the following will be the correct answer for problem number 16. I will wait for a minute, then I will check the chat box, okay? Let me check the chat box now. Let's see if there is any answer for problem number 16. Okay, number B is the answer which I have received. Let me check if number B is the correct answer or not. So the correct answer will be, it is number D. Isosteric groups can be used to determine whether a group is involved in hydrogen binding. This statement is true. Okay. Now let us understand what are the isostars and let us discuss it. Okay. So basically, isostars are the atoms or group of atoms which share the same valency and which have chemical and physical similarities. For example, SH, NH2, CH3 are the isostars of OH, S, and whereas the S, NH, and CH2 are the isostar of O. Isostar can be used to determine whether a particular group is an important binding group or not, by altering the character of the molecule as controlled way as possible. Replacing O with CH2, for example, makes a little difference in the size and analog, but it will have a marked effect on the polarity, electron distribution, and bonding. Replacing OH with a larger SH may not have such an influence on electronic character, but steric factor became more significant. Isosteric groups could be used to determine whether a particular group is involved in hydrogen bonding. For example, replacing OH with ACH3 would completely eliminate the hydrogen bonding, whereas replacing OH with NH2 would not. A beta blocker propanolol has a ethyl linkage. Okay, replacement of OCH2 with the isostar CHSCH2 CH2 CH2 eliminates the activity, whereas replacement with NCH2 retains the activity. So let us now see the option number 2, B and C. So number B, it says that replacement of OCH2 with SCH2 retains the activity, whereas the replacement of OCH2 with NHCH2 eliminates the activity. But it is the opposite way. Okay, replacement of the OCH2 with the SCH2, SCH or CH2CH2 eliminates the activity of propanolol, whereas replacement with NHCH2 retains the activity, although it is reduced. This results show that ether oxygen is more important activity of a drug and suggests that it is involved in hydrogen bonding with the receptor. So there are also, these are the examples of the how these bioisostars are important. So this is the aminopyridine. Okay, and this but in aminopyridine, this group is found to be have some carcinogenic effect due to this group. Okay, the bioisosteric modification by replacing this to this. So aminopyridine pyridine marketed as an analgesic or anti-inflammatory in 1922. It was revealed that it was a carcinogen, but propylphenazone was later developed, okay, by Roche in 1951, and bioisosteric modification of this dimethyl amino group is removed its carcinogenic action. So bioisostars are the substituent of the group of chemical, physical, and chemical properties which produce broadly similar biological effect of the in a chemical compound. In drug design, the purpose of exchanging one bioisostar to another is to enhance the desired biological and physical properties of the compound without marking significant changes to the chemical structure. Okay. Now I will go to the next problem. Problem number 70 says introducing a methyl substituent to the dopamine antagonist results in the dramatic reduction of affinity. What is the role of the methyl group here? Your options are number A, metabolic blocker, number B, configurational blocker, number C, conformational blocker, and number D, beta blocker. Introducing a methyl substituent to the dopamine antagonist results in the dramatic reduction of the affinity. What is the role of the methyl group here? Your options are it is acting as a metabolic blocker, number B, configurational blocker, number C, conformational blocker, and number D, beta blocker. Which of the following will be the correct answer for problem number 70? Let me know. A, B, C, or D, which will be the correct answer for problem number 17? Introducing a methyl substituent to the dopamine antagonist results in dramatic reduction of affinity. What is the role of the methyl group? I will check the answer after a minute. So let me know. Till then, A, B, C, or D, which of the following you think is the correct answer? Let me check the chat box. Okay. So there is no answer for this problem number 17. Let me check what is the correct answer and let us discuss it. So the role of methyl group will be it acts as a conformational blocker. Okay. So this is the structure number one and this is the structure number two. So here you can see the introduction of methyl group in this position. So another tactic has the same effect to use the conformation blocker. In certain situation, a quite simple substituent can hinder the free rotation of the single bond. So as you can see in this particular structure, this single bond can freely rotate. Okay, but to hinder the this rotation, what we can do, we can rigidify the particular structure. So rigidification is one of the ways. So another way is the introducing the conformational blocker. So introducing a methyl substituent on the to the dopamine D3 antagonist gives the structure to that results in the dramatic reduction of the affinity. So if you introduce a methyl group in this position, as you can see, as similar to this position, so now this single bond rotation will be drastically reduced and which results in the dramatic reduction in the affinity. In structure 1, the free rotation around the connecting bond allows the molecule to adopt a conformation where the aromatic rings are coplanar when and that is the active conformation of the receptor. So when this two rings are coplanar, that will be the active conformation. But when there is a methyl group here, now this two rings being coplanar will not be possible. So this particular conformation blocker or this methyl group rejects this active conformation. So in some cases, the introduction of this conformation blocker can increase the particular activity. But in this particular case, where this the introduction of methyl substituent to dopamine antagonist, it results in the dramatic decrease of the activity because for activity of this particular compound, these two rings need to be coplanar. But when methyl group is here, it is not possible for this two ring to be on the same plane. Okay, so that is why the particular affinity is reduced. Now let me go to the steric clash between the new methyl group and the orthoproton. So this orthoproton and this methyl group on the neighboring ring, it prevents both the rings being on the same plane. Free rotation around the bond between two rings is no longer possible. So the structure adopts a conformation where two rings are at an angle to each other, but this is not an active conformation and that is why the activity decreases. Okay. Now I will go to the next problem. Problem number 18 says which of the following statement is false regarding the lactams? Your options are number A, bioisosteric replacement, number B, ring fusion, number B, ring simplification, and number D, ring expansion. Okay, I think there is some issue with this question. Okay, no, it is fine. So, which will be false regarding the lactams? Okay, I think there is some issue with this question. Let me just directly go to the answer part because there is a issue with the drafting of the question. Okay. Okay, so basically, in this case, okay. Oh, okay. So I just wanted to change the question from 1 second. Okay, so sorry, the question will be like this. Let me just correct this thing. Okay, sorry. So I just wanted to I had to change the question because there was some issue. Okay. Okay, let me just show you one more time the question. So the question just that I intended to give you, like, which of the following strategies is used to achieve the procaine from cocaine? Okay, so your options were bioisosteric replacement, ring fusion, simplification, or ring expansion. So the correct answer is simplification. So what is simplification? So basically this is the structure of the cocaine and this is the structure of the procaine. Okay. So here the pharmacophores are given in the blue color. So this phenyl ring with this C double bond O means this particular ester bond, and here we have this this carbon and this your nitrogen. So all these blue portions are the pharmacophore of the cocaine. So for the development of cocaine from procaine from cocaine have used the strategy of the simplification. Okay, so simplification tactics have been used successfully with the alkaloid cocaine. Cocaine has a local anesthetic properties that simplification led to the development of the local anesthetic which could be easily synthesized in the laboratory. First earliest was the procaine or novocaine. This discovered in 1909. Simplification tactics have proved effective in the design is much simpler morphine analog. So as you can see, this cocaine has some difficult, the structure is very complicated. As you can see, there is the 6-member ring with fused with a 5-member ring with the nitrogen. Only thus having this NET2 serves the purpose. So this is the example of the simplification. Simplification tactics was also used in the development of devazepide from the microbial metabolite asparlysine. So this is the asparlysine and this is the devapazide which has the particular simplification of this asparlysine. As you can see, again blue color portions are the pharmacophores and the other groups, if it is removed, also their activity retains. Okay, the benzodiazepine and endoskeleton inherent the asparagine are importantly for activity and have been retained. Both the asparagine and devazepide acts as an antagonist and neuropeptide chemical messenger called cholecytokinin, which has been implicated causing the panic attacks. Therefore, antagonist may be used to treating such attacks. Okay. So the question was this one, like I wanted to give the question regarding simplification. So simplification is a very good tactic also in the case of optimizing the drug target interaction. For example, like other like your conformation blocker or rigidification or extension strategies. Okay. So problem number 19, it says which of the following statements is false regarding the binding role of the alkenes. Your options are number A, they are flat and hydrophobic. Number B, they, like aromatic rings, can interact through the van der Waal interaction. Number C, they interact with the binding site less poorly compared to the saturated analogues. And number D, they are easier to reduce compared to the aromatic rings. Which of the following statements is false regarding the binding role of alkenes? Your options are number A, they are flat and hydrophobic. Number B, they, like aromatic rings, can interact through the van der Waals interaction. Number C, they interact with the binding site less poorly compared to the saturated analogues. And number D, they are easier to reduce compared to the aromatic rings. Which of the following statements is false regarding the binding role of alkenes? Let me know A, B, C or D. Which of the following will be the correct answer? I will wait for a minute or two and then I will check the chat box. Which of the following statements is false regarding the binding role of alkenes? Your options are number A, they are flat and hydrophobic. Number B, they, like aromatic rings, can interact through the van der Waals interaction. Number C, they interact with the binding site less poorly compared to their saturated analogues. And number D, they are easier to reduce compared to the aromatic rings. Which of the following statements is false regarding the binding role of alkenes? Let me check the chat box. Okay, number C, I have received only one answer saying that number C is the correct answer. Let me check if number C is the correct answer or not. So the correct answer will be yes, number C, they interact with the binding site less poorly compared to their saturated analog. This statement is false regarding the binding role of the alkenes. So, like aromatic rings, alkenes are planar and hydrophobic. So, they too can interact with the hydrophobic regions of the binding site through the van der Waals interaction. The activity of the equivalent saturated analog would be worth testing, as the saturated alkyl region is bulkier and cannot approach the relevant region of the binding site so closely. Alkenes are generally easier to reduce than aromatic rings, so it may be possible to prepare the saturated analog directly from the lead compound. So as you can see, this alkene, it is flat and it has a better binding hydrophobic interaction, whereas the alkyl ring, which is bulkier, and it does not have very much good binding in comparison. So now let us see the structures. So the options, the number A is, it says they are flat and hydrophobic, which is true. Number B, they like aromatic ring can interact through van der Waals interaction. This is also true. Number C, which says they interact with the binding site less poorly compared to the saturated analog. It is this is untrue. This is false. Okay, because they can interact with the binding site better compared to the saturated analog. And number D, they are easier to reduce compared to aromatic ring. This is also true. Reducing an aromatic ring is more difficult than to reducing to the alkene. So when we have alkene, it is possible to prepare saturated analog directly from the lead compound, whereas in case of the aromatic ring, if it is not that much simple. Okay, and as this figure, it is clearly shows how the particular saturated analogs, they bind poorly than the alkenes. Okay, now, the last question of today's session, it says which of the following statements is false regarding the quaternary ammonium group. Your options are number A, the neurotransmitter acetylcholine contains a quaternary ammonium group. Number B, quaternary ammonium salt interact with the carboxylate group through the hydrophobic interaction. Number C, quaternary ammonium salt interact with the phenyl rings of the binding site through the induced dipole interaction. Number D, none of the above. Which of the following statements is false regarding quaternary ammonium group? Your options are number A, the neurotransmitter acetylcholine contains a quaternary ammonium group. Number B, quaternary ammonium salts interact with the carboxylate group through the hydrophobic interaction. Number C, quaternary ammonium salts interact with the phenyl rings of the binding site with the induced dipole interaction. Number D, none of the above. Let me know A, B, C or D, which of the following will be the correct answer for problem number 20. Which statement is false regarding the quaternary ammonium ion? Which of the following statements is false regarding the quaternary ammonium ion? Number A, the neurotransmitter acetylcholine contains a quaternary ammonium group. Number B, quaternary ammonium salts interact with the carboxylate group through the hydrophobic interaction. Number C, quaternary ammonium salts interact with the phenyl rings of the binding site and through the induced dipole interaction. Number D, none of the above. Let me wait for another minute, then I will check the chat box. Which of the following statements is false regarding the quaternary ammonium group? Okay, let me check the chat box. Okay, I have received the answer D. Let me see if D is the correct answer or not. So the correct answer is number B, the statement which is false. It says that quaternary ammonium salts interact with the carboxylate group through the hydrophobic interaction, which is clearly not possible because quaternary ammonium salt and the carboxylate group, we both are the charged. So hydrophobic interaction cannot be possible in this case, right? So the quaternary ammonium salts are the ionized and can interact with the carboxylate group with the ionic interaction. Another possibility is the induced dipole interaction between the quaternary ammonium ion and any of the aromatic ring of the binding site. The positively charged nitrogen can distort the pi electron cloud of the aromatic ring such that a dipole is induced, whereby the face of the ring is slightly negative and the edges are slightly positive. This allows the interaction between the slightly negative faces of aromatic ring and positive charge of the quaternary ammonium ion. This is also known as the pi-cation interaction. The importance of this interaction could be tested by synthesizing an analog that has a tertiary amine group rather than a quaternary ammonium group. Of course, it is possible that such group could ionize by becoming protonated in the interact with the same way. Converting the amine to amide would prevent this possibility. Neurotransmitter acetylcholine has a quaternary ammonium group which is thought to bind to the binding site of its target receptor by ionic bonding or induced dipole interaction. So for example, so this is the drug with the carboxylate group. When it is present in the binding site, the quaternary ammonium ion interacts with the ionic interaction, whereas in case of the this aromatic rings, it induces a dipole where the center of the ring is slightly negative and the edges are positive. So it is an induced dipole or a pi-cation interaction. Okay. So statement number B, which says that the quaternary ammonium ion salts interact with the carboxylate group through the hydrophobic interaction, it is not true. It is the ionic interaction. Okay. So this was the last question of today's session. So today, basically what we have discussed, so today mainly we have discussed about various tactics by which the drug target interaction can be optimized, like rigidification, conformation blocker, then we have the simplification technique, extension, or this kind of techniques can be used, and also introduction of this, for example, this alkene as the binding group, the quaternary ammonium salts, all these are the examples by which the optimization can be takes place. Okay, so I will share my email ID now. If you have any doubt or any query regarding today's class or any previous class, please let me know via email, or you can put your queries in the this SWAM portal's discussion forum. You can put your queries. The discussion forum is here in the question answer. Where to find this particular problem solving session material, I have already discussed. Okay, so you can check it later. So if there is no query, I will close the session for today. So thank you for answering and joining the class and answering. We will meet on next Monday with our week 9 live session. Okay. Till then, if you have any problem or any doubt, please let me know via email. I will try to discuss that particular problem in the next upcoming class. Okay. I am closing the session for today. Thank you.